And so last week you guys had your test, and it was about mitosis and other topics from earlier on in the weeks, the months, I guess month even. So what I want to do today is first say welcome back from, and I hope you had a great Veterans Day. But now we're gonna go into some new information, maybe not new to you, or some of you may have had experience with these words and terms and topics before, but it's important that you revisit it if you have learned it before, but also for those people that haven't, this is your opportunity to learn about what it is meiosis. Okay, so before I get into meiosis, I do think it's really important to do a little review of mitosis so that you guys are aware of what the difference really is between the two of them. They are specific to certain organisms, and as particularly meiosis. So just please don't get them confused. If you ever need clarification, you can send me an email now, since I've returned from my limited access of email. So if you do have any questions, send me a message on Remind. I will be available to respond. But let's move on. So we have to talk. We can't talk about mitosis unless we talk about the cell cycle. So the cell cycle includes all the phases of a cell, basically. And so in this case, you see interphase. You see interphase is taking up actually most of the cell's lifespan. And so what it does is the interphase, there is cell growth that's happening, so it's getting larger because it needs to make more organelles. And there also needs to be DNA copied in interphase, and then the cell will also kind of have checkpoints and see if everything's good to go for when mitosis actually happens. And if you notice, mitosis, or the mitotic phase, is actually the smallest part of the cell cycle. So most cells spend their time in interphase. Okay, so it also mentions how mitosis is involved in the formation of two daughter cells. That's important. Daughter cells are just basically what is produced. Okay, I don't know why they don't call it son cells versus daughter cells. Doesn't really matter. The point is that you get two identical daughter cells in the process of mitosis. Okay, so here are the 5 stages. Just a review again, most of the cell spends its time in interphase, but there's a lot of things still happening at that time. Copying of DNA, copying of organelles, all that stuff, getting ready for the process of mitosis. And so we have PMAT, prophase, where you see the condensing of a chromosome, so you're actually able to visualize them. Here you can't really see them. In prophase you can. So they're getting ready to line up. So what happens is that the chromosomes will line up in the middle of the plate, of the metaphase plate, it's called. And then what will happen is that in anaphase, the centrioles and microtubules, they're just like little, could think of them like hooks or lines that catch on to the chromosomes, pull the sister chromatids apart. Right here you can see each of them getting pulled apart across the cell. And then telophase, which leads eventually to cytokinesis. In telophase you have a whole new a cell, basically. Not yet, but there's this indentation happening, but you end up having a new set of identical DNA in both cells. And in cytokinesis means actually cell cutting. And so what happens is the cell breaks in half and makes the 2 daughter cells. Okay, important to know about mitosis is that it occurs in most of our somatic cells. Another word, what somatic cells are body cells. So our skin cells, our cells in our liver, the cells in our stomach, or, all those places, everywhere that you can think of, are our body cells. They're different from our sex cells or our gametes, but mitosis is exclusive to somatic cells. So in mitosis, we get a clone of the cell before it, so you'll get 2 identical daughter cells for the most part, unless mutation happens, and that can always happen. but most of the time it's getting identical daughter cells. Also, mitosis is important to understand because this type of cell division is how asexual reproduction happens in certain organisms. So, for example, here is a picture of amoeba. And so the term binary fission is just a type of asexual reproduction. There's a few other types as well, but the same product happens. You get the same product. You get two identical daughter cells. So this was your original amoeba. It goes through the process of mitosis, and you get the two, or you could say asexual reproduction, and you get the two daughter cells. Okay? So I hope that makes sense about the whirlwind. I know I gave you a little bit of whirlwind about mitosis, but by this point you really should know what it's all about. Now, here's meiosis. Hopefully you can recognize that this is a chromosome. Maybe you've seen that before. It's a picture of a chromosome. This is one sister chromatid. Here's another sister chromatid. Okay. And we'll talk a little bit about that in a second. So meiosis is it's called reduction division. What happens is actually you get a reduction in the amount of chromosomes, whereas in mitosis you don't. In mitosis you keep the amount of chromosomes. Also in mitosis you will see a lot of genetic recombination. That's really important in sexually reproducing organisms, having genetic diversity, meaning different types of genes. It's what makes us all humans, but that we don't look all the same is because of that genetic recombination. Okay, so this is quite a bit of stuff. So meiosis is a form of cell division, but we have gametes that are formed in the process. Gametes are our sex cells. So for example, our sex cells would be sperm or egg. Okay, and so these gametes, these sex cells, have half the number of chromosomes than a normal body cell. Okay, and so a normal cell, body cells, are considered diploid. Okay, so they have 2 times N. Okay, so it's a little bit of math in a way. N stands for number of chromosomes. So this isn't just for humans, but if maybe you've heard is that humans have 46 chromosomes, right? So if a regular human body cell has 46 chromosomes, then 2N would equal 46. So if you divide through by that, you get N equaling what? 23. So if you divided 46 by 2, you get 23. And so in meiosis, what you get is diploid, a normal diploid cell becoming haploid through the process of cell division. It'll become, so instead of having 46 chromosomes, at the end of meiosis, you'll see 23. Now that's in human sexually or human reproduction, right? And human meiosis, if you wanted to say it that way. Other organisms have different amount of chromosomes, but this equation still holds true. In meiosis, you will see a diploid cell becoming haploid through the process of meiosis. It'll be just a different amount of chromosomes. So meiosis is involved in sexual reproduction. Okay, so that's really important to understand. You will never see meiosis in asexual reproduction. And it makes sense because in meiosis you get so much diversity, whereas in mitosis you're getting basically cloning. Now, be aware that humans do both undergo meiosis and mitosis when it comes to wound healing or fixing a cut basically, or growing in general. A lot of mitosis is occurring. Okay, so it's happening. Remember, it's happening in all our body cells. I'm sorry. Let me go back. And so meiosis is broken up into 2 phases, kind of. We have meiosis 1 and meiosis 2. You'll find that meiosis 2 is very much like the process of mitosis, but you'll see in a few minutes what makes meiosis different from mitosis. Okay, again, sex cells. These are sex cells. They're called precursor sex cells with the 46 chromosomes. Will divide and then they produce gametes. These are our matured gametes. Okay, so sperm and egg are considered mature gametes. Gametes will always have half the number of chromosomes as the body cells do, and we have meiosis occurring in our gonads. Those are sex organs, basically. So in that case, it's our testes or our ovaries. That's where we will see testes will produce sperm, and ovaries will produce eggs. So the term for the production of sperm, genesis, means growth, right, or new. So genesis, the growth of new sperm, happens in the testes. The name of the process is called spermatogenesis. In females, it's called oogenesis, and it's the production of an egg or ova. So another word for egg is ova. Okay. Okay. So this is what it looks like in spermatogenesis. This is just very simplified, right? We will talk more about the phases of meiosis or the process of meiosis in a minute. But in spermatogenesis, this is a type of meiosis. You will get this precursor human sex cell at 46 chromosomes. Then will have 20. Will end up having 23 chromosomes at the end of meiosis 1, and then there will be division happening at meiosis 2. And this is why I say that you still end up with 23 chromosomes. It doesn't half again, but we get 4 haploid daughter cells in meiosis. Okay, so 4 haploid sperm daughter cells, doesn't matter how you want to say it, but this is a production of sperm or just what happens in meiosis. Again, overly simplified. Okay, oogenesis is a little different. You still end up having 4 products, but what happens is there's only one viable egg that is produced through meiosis, and the rest of them become polar bodies. These polar bodies can help. They basically give away their covering, their outer covering, and then of course they die because nourishment, because they want to give that one egg the ability to produce a viable offspring when the time comes for it. Okay, so you end up getting one good egg, and the rest are polar bodies, which eventually end up dying and being reabsorbed. Okay, so interphase 1, it just kind of looks like mitosis interphase. You see DNA replication happening, and then you'll get those 2 identical sister chromatids, right? That's what's happening in interphase 1. Okay, they're attached to the centromere. You could remember centromere, center. Okay, and so this is what happens at interphase 1. You can't really see the DNA, right? It's going to be kind of packed, not packed, they're loose actually. Chromatin is basically DNA being all unwound and really hard to see under the microscope. You can't see it in interphase. That's the same thing as mitosis. You can't see them in interphase. So in interphase 1, you can't see that. So everything is pretty much intact in interphase 1. Again, nucleus and nucleolus is visible. So now we start meiosis 1. And so, quick overview. This is the first cell division that happens, and it reduces the chromosome number by 1/2. That's important, and we'll discuss why we need to cut the chromosome number by 1/2 in meiosis in sexually reproducing organisms. So we have 4 phases in meiosis 1. We have prophase 1, metaphase 1, anaphase 1, and telophase 1. So we'll go through those right now. Okay, so in prophase 1, not completely unlike prophase in mitosis, the chromosomes condense, meaning that you can see them. They become tightly wound. And then we have something called synapses happening. So we have homologous chromosomes coming together to form a tetrad. So if you would say, so what you would do is you would have that basically chromosomes that are alike will match up with one another. Okay, and then they form something called a tetrad. And so it's basically 2 chromosomes. So this is 1 chromosome, this is another chromosome, and then joining up together. And that's the tetrad. But understand, since there's 2 chromosomes, right? You have 1 and then you have the 2. We have 4 chromatids, okay? So there's 1, 2, 3, 4. That's 4 chromatids. 1, 2, 3, 4. And what you'll see is that you need to be able to distinguish between sister and non-sister chromatids. So in this case, you have both the blue are considered sister chromatids, right? They're still attached to each other. That so you can think of like sisters being attached at the hip, right? So sister chromosomes are attached to each other. Non-sister chromosomes are basically how the two chromatids. I'm sorry, they're the two chromatids matching up with one another. So they have like areas. Okay, so it would be basically this chromatid and this chromatid, right? So the blue chromatid and the pink chromatid would be considered non-sister chromatids. Okay, again, this is important to understand. This is why there's another slide for it. Okay, so another word for non-sister chromatids are homologs. They are homologous chromosomes. Okay, so homologous chromosomes have DNA that codes for the same genes, but different versions of those genes. Okay, so they're not identical to one another. While sister chromatids, right, the ones that are attached to each other, are exact replicas, the homologous chromosomes are not exact replicas. And so here's how I can explain this a little bit better. So you have, say, this dark purple would code for, let's say, eye color. Okay, so if the purple codes for eye color, the dark purple, what you could think of it as is the dark purple would be, let's say, blue eyes. Okay, and then they'll say the light purple, that lavender, is codes for green eyes. Okay, so those are still coding for the same gene, right? Eye color, but you're getting two different results, right? That they're just different versions of that gene. That while they both code for eye color, this will be blue, right? The dark purple will be blue eye color, and the light purple will be a green eye color. So we can say, so understand that they share DNA, or rather, the two dark purples will share the same exact DNA, but then when we talk about the dark purple and the light purple, they are just different versions. Okay. Okay. So we have two chromosomes coming together, forming this tetrad. That's a process of synapsis that only occurs in prophase 1. Okay, those are our homologous chromosomes. Here you go. And then we have each chromosome themselves have their own sister chromatids. You could think of them being as attached at the hip. Okay? And they form this tetrad. All right, so homologous chromosomes, they're a pair of chromosomes, maternal and paternal, that are similar in shape and size. So if you'd like to go, actually what I think I should do is I'll go back in a second, but understand that the chromosomes come from mom and dad. Okay, so they're similar in shape and size. So if we're gonna go, let me just go back real quickly let's go to this picture. Let's say the chromosome on the left came from mom and the chromosome on the right came from dad. And we could say mom had blue eyes, so she passed that on to you, and dad had green eyes and he passed that on to you. And then that's how we get these homologous chromosomes matching up in meiosis. Okay, so let me just move forward a little bit. Okay, there we go. So humans have 23 pairs of homologous chromosomes. We have autosomes, which are for our basically coding for our body, all the cells of our body, all the traits of our body. So the first 22 pairs are autosomes. The last pair are our sex chromosomes. So that's XX and XY. Okay, so and what is this talking about here? The loci or locus is just an area of DNA that might, match up with another on the homologous chromosomes. Okay, so here, like just what I mentioned earlier. So that may be that this homologous chromosome, these two, in this tetrad, say this, the pink is mom and the yellow is dad, coming from dad. So we can say mom had blue eyes, and the eye color locus for the mom is located in that one section, the loci locus of that chromosome, that one area of the chromosome, and that's the same for the dad. But in that case, it's probably green, because we're just going to keep going with that same example. And then on another part of the chromosome, you may have coding for hair color, and so mom might have brown hair and dad might have black hair. Okay, so just understand that, it's not just a whole chromosome that codes for one thing. That's not how it works. It's areas of DNA, right? Sections of DNA throughout those chromosomes. Okay, so okay, so this is saying it's from dad and from mom, but you can. It, I'm gonna go from my example, so there's no confusion. Okay, so crossing over is really important. Okay, so the reason being is because it causes genetic recombination. That gets the diversity that we so need for the survival of most species. Okay, so crossing over can actually happen between non-sister chromatids. So those are our homologous chromosomes at sites called chiasmata. Okay, or the chiasma. That's more than one. So in crossing over, we have segments of the homologous chromosomes breaking and reattaching to the other chromatid. Okay, and so in the chiasmata is where the chromosomes actually are sitting on top of each other, and that's where the exchange of genes happens. That's that process of crossing over. All right, so here's a picture of that. Okay, so of course we result in variation from the process of crossing over. So this is basically saying right here, chiasmata is a site of crossing over. So right here is where we get that site of crossing overs, where you see them kind of like intertwining um, homologous chromosomes. And since they're homologous, nothing messes up, right? Because they're still coding for the same stuff. That's why they can go ahead and do that. And so then you see a switch happening in the homologous chromosomes, and so boom, mom gave the hair color, maybe let's say, and dad gave his hair color. Okay, so that's how that works. Okay, so this is the process of beginning process of synapsis is when we have the pairing of homologous chromosomes, but then crossing over is happening at this chiasma or chiasmata, and then we have genetic recombination happening. Okay, now like I mentioned before, we have 22 autosomes, but we have one pair of sex chromosomes. So if you have an XX chromosome, you have a female. And then if you have XY, so XX are obviously they're going to look the same. And then if you have XY, the Y is going to be a little bit smaller, but it's still going to match with the X. Okay, but they call it XY because it actually looks more like a Y in karyotyping. Um, so yeah. All right. So in meiosis one, you end up having this is a great picture. You end up having your homologs separating, and so this in this process you can have crossing over, right? That adds to recombination or genetic diversity. So let's move on. Okay. So in prophase one, let's see, there we go. The nucleus and nucleolus disappear. You get the chromosomes coiling up, so condensing. The pairing happens between the DNA or the homologous chromosomes, and then tetrads form, and then that's where we get the crossing over, where we can have that diversity. Okay. Then we have metaphase one. So metaphase, same idea. Meta, just remember meta mean middle. You could remember M for middle. And so these tetrads will align at the equator, so the center. And in here, this is where independent assortment happens. So when the process of separation happens, it happens randomly, okay? And so you'll get genetic recombination this way because anything can happen in this point. When it starts to get pulled apart, you can have different versions of the gene uh, forming all these different combinations. That's what independent assortment. There's no rhyme or reason to it. It's not. There's no system for this. It kind of just pulls it apart randomly. And so here, this is basically we're getting into genetics here. So if you want to talk about A being coding for eye color, B codes for hair color. What you can say is that A is blue eyes, big A is blue eyes, little a, let's say big A is brown eyes because that's what it really is in genetics. Big A is brown eyes, little a is blue eyes, and we'll say big B is brown hair and little b is blonde hair. So what'll happen is you get all these combinations that can happen, right? You can get somebody with brown In this case, it would be brown eyes and brown hair, brown eyes, brown hair, the big A, big A, big B, and then if you have little A, little B, you get we'll say I believe we were just talking about it being blue eyes, right? Yeah, little A is blue eyes, so it would be blue eyes, and little B would be blonde hair, right? Blue eyes, blonde hair. But right, you can also get this combination too, right? So you can get brown eyes and um, blonde hair, right? Or it says blonde, brown eyes and blonde hair, and or you get blue eyes and brown hair. Okay, so that's all those combinations. You get so many different combinations, which also adds to the diversity that we see. Okay, sorry, I just want to show this real quick. It's the same thing. Homologs are gonna line up the equator or the metaphase plate, and then they will start to separate. But that'll happen in anaphase one. So in independent assortment, there are many different combinations that a human male can produce. So doing the math, you can get, if you have 2 to the n, that's the formula to get all those combinations. We know 2n is, our n is 23, right? We just mentioned that earlier. So 2 to the 23 combinations, that's 8 million combinations. There's the diversity we're looking for. Okay, so in anaphase 1, separation happens between the homologous chromosomes. They move towards the poles. But sister chromatids are still together in their centromeres, right? They're still attached to the hip. Okay, so here's a picture of that. Getting pulled away towards the poles, the homologous chromosomes get pulled. Okay. Telophase 1. Now each pole has a 1n or just n set of chromosomes. And then we have cell cutting happening, and then we have the daughter cells that are formed. Now, for humans, in this case, you have 23 chromosomes, okay, in each daughter cell, right, at the end of telophase 1. So that's just the picture being shown here. Telophase 1 is the indentation that happens, and inside of meiosis is when you get the 2 daughter cells, okay? So in meiosis 2, now we're having the sister chromatids separate. So we had the homologs separating in meiosis 1. Sister chromatids are now separating in meiosis 2, okay? This is why I say it's a lot more like mitosis, right? And it says it right here. There's no interphase. If there is, it's very short. Really, it's negligible, not really important to know. But it is important to understand that meiosis 2 does have similar hallmarks to mitosis, okay? So prophase 2 is the same as prophase in mitosis. You end up having the nucleus and nucleolus disappearing, the chromosomes condensing, and then you get your spindle, and you can start to see that they start to attach at the centromeres. In metaphase 2, you get the same as metaphase in mitosis. Now it's the chromosomes lining up in the center, not homologous chromosomes, just the sister chromatids as their full chromosome being lined up at the equator. And of course, predictably, in anaphase 2, you will get the sister chromatids separating. They start to get pulled to the opposite poles, the opposite sides of the cell. That's in anaphase 2. Okay, same as the anaphase in mitosis. Then telophase 2, same as telophase in mitosis, right? You will get that indentation. You will get 4 haploid daughter cells. So again, 4 cells, haploid daughter cells that have 23 chromosomes in them, okay? So that's for humans, of course. Now, this little picture is just showing fertilization. This is why meiosis is so important, okay? You definitely need to have the amount of chromosomes. Why? Because when you have sex cells, if you had 46 chromosomes, then when fertilization occurred, which is when a sperm meets the egg, you would have way too many chromosomes inside, right? A normal human body or normal human cell should have 46, but if say a sperm had 46 and an egg had 46, you'd have much more. Okay, you'd have way too much. So that's, what is that, 90, 46, is that 92? Yeah, I believe it is. If I can't do the math, but regardless, you need to understand is that this is why we need to have that division happening, the reduction division. We need to bring it down to 23 so that the 23 from the sperm can fertilize the 23 in the egg, and then we get our beautiful number of 46 for our zygote, which is a zygote. Another word for zygote is a fertilized egg. Okay, so telophase 2, we get our sister chromatids separating to form the 4 haploid daughter cells. And so this is just a quick picture of what's happening in telophase. In meiosis. This is the beginning. You saw the crossing over that happens. Now it starts to go so fast. But that was in meiosis 1. Now this is meiosis 2. And you can see how the sister chromatids are separating. Okay, so I'll just go through this one more time so you guys understand. Again, this is a process of meiosis 1 right now. Okay, we got our homologous chromosomes lining up right there, crossing over, happening in prophase 1, and then we split them up. And then meiosis 2 starts, and you start to see it occurring the same way as mitosis, in that sister chromatids separate, and then we get our 4 haploid daughter cells. Okay, so again, meiosis is very important. It provides variation. Another term I've been saying throughout this lecture is genetic recombination. That's important to the population because it is involved in natural selection. Of course, we're not getting into natural selection right now, but that variety allows for organisms, not just humans, to survive in their ecosystem. Okay, so the most fit, right? The one with the best genes will survive to reproduce and then pass on those genes, pass on those traits. Okay, and so a karyotype is what humans do this for when we look at. There's geneticists that look at this to understand what chances are of children developing any disorders for anyone that might be carrying a genetic disorder. So they look at karyotypes and they look at the organized picture of chromosomes arranged in pairs by size. Okay, so the pairs 1 through 22 are our autosomes. They code for everything in the body from hair color to eye color to height to anything you can think of. And skin color, right? And those are our autosomes. The last pair are sex chromosomes, and hopefully you're able to figure out that in this particular karyotype, this is a male because you see the X chromosome and you also see the Y chromosome. Okay, and then in this case, you can see they're both X, so hopefully you're able to figure out that that is a female. Okay, let's see if there you go. And now this is showing a example of trisomy 21. Maybe you may have heard of that before in terms of Down syndrome. What's happening is that there ended up being 3 chromosomes in 21. Okay, and so you end up seeing some results resulting in Down syndrome. Okay, so that is the end of my PowerPoint. I just want to make sure that you guys understand what is going on in meiosis. If there's any questions, again, please do not hesitate to email me. We will move forward from this, and hopefully it is clear to you now what meiosis is all about. If you did not, if you were having trouble knowing the difference between mitosis and meiosis.